We're going to do our liver lab material today, and we're going to begin with just a few drawings here on the board, and then we'll go to the microscope and we'll show you those images from the microscope up on the screen, on the television screen. So to begin with, our basic shape of our liver here, on the outside of the liver you're going to have a thin layer of connective tissue, and on the outside of that a mesothelial layer. So if you were to take a little tiny chunk of that outside layer and look at it under the microscope, you would see that it consists of a lot of collagen fiber out here, and among those collagen fibers, of course, there would be a fair amount of other connective tissue related cells, mostly fibroblast cells in there. And then on the very outside, you would have that layer of mesothelium, simple squamous cells with fairly flattened nuclei there on the outside of the capsule. So in here you would find nuclei of fibroblasts among the collagen, and then the mesothelium on the outside. So we have a special name for this outer capsule to the liver, and we call it Glisson's capsule, G-L-I-S-S-O-N'-S, Glisson's capsule on the outside of all the liver tissue. So the main hepatocytes and other liver tissues would be deep to that. So we have this entire liver covered with Glisson's capsule. Coming up on the underneath side of the liver here, remember we have a location where blood vessels will enter into the liver and then branch out and go their different directions to get to every part of the liver, to supply the liver with all of the blood in there. And there's another vessel carrying portal blood. I'm going to put it in blue, but we usually do try to do it in a yellowish color, but this will show up better. Hepatic portal vein blood is going to come in here, and it's going to follow the same path as the arteries, the hepatic arteries that bring blood in here. So everywhere you have a hepatic artery branch, you'll have a hepatic portal vein branch to follow it. At the same time, there are bile ducts that lead out the opposite direction. Bile is usually indicated in green, so here's a green line going out. So this is going out this way. And every place you have a hepatic portal vein branch, a hepatic artery branch, you also have a bile duct that leads out and connects and goes toward the common bile duct, which leads into the small intestine. So we'll next be looking at the portal triad and the tissues associated with that, and then we will look at the liver lobule structure. Here we have a crude drawing of portal triad material here, just to represent what's going on in this. Portal triad, it consists of a hepatic artery branch, a hepatic portal vein branch, and a bile duct. Now, not all of our portal triad locations consist of only one artery, one vein, and one bile duct. Sometimes there'll be 2 or 3 bile ducts associated with a single portal vein branch and artery branch. Sometimes your arteries branch before you get to a next branch of your portal vein, so there could be a couple or 3 arteries before and a single portal vein, or reverse to that. So anyway, the exact number is not that important. It's important that you have all 3 represented to see it as a portal triad in there. So this here is a simple cuboidal epithelium around your bile duct. So this is our bile duct of our portal triad. Over here we have an artery with these blue nuclei around it are to represent smooth muscle nuclei. But anyway, this is our hepatic artery branch. And this larger structure, and it's always, it seems, much larger than the artery in the portal triad. This is the hepatic portal vein branch. So we'll be looking in our sections for representatives that show all these features. hepatic artery branch, hepatic portal vein branch, and a bile duct branch in there. And those are always associated with a fair amount of connective tissues that surround that. So there will be collagen, and I'm going to put it in brown, this reddish brown color here. But anyway, there would be a fair amount of collagen surrounding this whole structure, all of it together. Be a fairly well defined region of connective tissue surrounding our portal triad tissues. And in there would be fibroblasts, of course, along with all this collagen. So that's a portal triad. They can be large, they can be small, depending on how far you are down that branching pathway within the liver. The larger the bile duct is, the more columnar the cells will be. So closer to the porta hepatis, where it exits the liver, you're going to have a taller epithelium in your bile ducts there. Where you see the taller bile ducts, you'll probably see a larger artery and larger vein to go along with it. Next, we're going to look at the central vein and radiating plates of hepatocytes in the liver lobule. In this picture, I've got the central vein here, and around that central vein, you're going to have plates of hepatocytes that radiate out like spokes of a wheel, and then there will also be the sinusoids, the blood sinusoids that lead into the central vein, coming in from all directions around it. So there's a very distinct radial pattern to a liver lobule. Out at the very outsides of the lobule will be locations with these portal triads we just talked about. So your blood supply leading into these sinusoids, that blood comes from both hepatic portal vein and hepatic arteries that feed the blood in toward the center. There will be simple squamous cell nuclei, these endothelial nuclei, on the inside of our central vein, so that will be an easy to spot feature of our central vein, the simple squamous cells. Those simple squamous cells, endothelial cells, radiate out into these sinusoids out here. And also in your sinusoids, you have a special cell type that is intended to be a phagocytic cell. It's to clean up the tissues here as well. So in here you're going to find a few of these cells that are called Kupffer cells. Kupffer cells. So we've got 3 Kupffer cells drawn in. We've got our endothelial cells. These are hepatocytes. They have a large round nucleus, each of the hepatocytes. They're large, squarish kinds of cells, although they can be irregularly shaped in some positions. They don't form very good squares. For the most part, they're side-to-side cuboid-like cells that radiate out from that center and go along the length of each of the sinusoids out here. Now there's a gap between the endothelial cells of the sinusoids and the hepatocytes out here, known as the space of Dicey. The space of Dicey. So these sinusoids are somewhat, they're discontinuous capillaries. That means that they don't seal the fluids inside of that sinusoid space, but fluids can leak through into that gap between the sinusoid and the hepatocyte. So that little gap in there is the space of Dicey. That's where most of the physiology occurs, where you have interplay between the blood fluid and the liver cells, which is where the liver does its work. So we'll be looking at those things: central vein, sinusoids, endothelial cells, Kupffer cells, hepatocytes, the space of Dicey, and then we'll be looking at the bile flow in the other direction. Between the hepatocytes, these can be very difficult to find. I've drawn them in here as very obvious features. Here where there's a gap between the cells, these are bile canaliculi. This hepatocyte produces bile and secretes it into this tiny little space here, that bile canaliculus. Bile canaliculi come together and those secrete into a pathway that leads eventually into a bile duct out at the perimeter. Often before we reach the perimeter, however, we get into a place where there are cells that form a boundary around this flow path for the bile. This is a bile ductule, also known as the canal of Herring. Canal of Herring. So these are small cuboidal-like cells. They kind of form that stepping stone look like we saw in some intercalated ducts in the salivary glands. But anyway, these small bile ductules that carry bile after it leaves the canaliculus, heads through tiny spaces to enter into this duct and then eventually into a bile duct of the triad out at the perimeter of the lobule. So those are things we'll be looking for in the liver lobule structure. Here we are looking at our first slide showing part of Glissen's capsule on the outside of the liver. So we have a section over here that's all collagen. This is all hepatocytes and liver tissue here. This section through here is all collagen, and these cells through here, especially these dark narrow nuclei, these would be fibroblast nuclei. And on the very outside we have some nuclei here. These would be those simple squamous cells of the mesothelium, the mesothelial layer on the outside of the liver. All that together makes up Glissen's capsule. So there's our first view of Glissen's capsule. Now bring up another picture of Glissen's capsule. Going another way, here we have those simple squamous nuclei indicated over here, and about that much of it is Glissen's capsule here. So not as thick as it was in the previous example. Here's an artery, a prominent artery over here on this side of the screen. And all of this down here is liver tissue, hepatocytes and sinusoids and all that. A similar image. Again, we can see our collagen very well up in here. Looks like a dense irregular connective tissue type of collagen here. Simple squamous cells on the surface. And we kind of lose that connective tissue about here, and then down here, these are hepatocytes and liver tissues. Next, to look at some triads, portal triads. This is a fairly small portal triad. Now, not all the liver slides look the same. Some are darker staining, some are lighter staining. So there are better slides and not so good slides. But anyway. Right around here we can see the perimeter of this portal triad group. Here we have a ring of cuboidal cells making up the duct. Down here we have an artery branch, a branch of the hepatic artery. The nuclei of endothelial cells pushed to the middle toward the lumen there, and then we have some smooth muscle nuclei surrounding that. Over here, this larger space, these are all red blood cells here. A few other cell types and white blood cell types in there, but anyway, this large space is the hepatic portal vein branch. So everything around the artery, bile duct, and portal vein in here, these are all connective tissues associated with the portal triad. Outside of that, these are all hepatocytes, sinusoid spaces, the white spaces, and the darker cells with the nice round nuclei. These are all hepatocytes. Next example of a portal triad. This is a very large triad example. This is a lower magnification to get everything on the screen. But all this white space here, this is all the hepatic portal vein branch. There are a couple of arteries, fairly large arteries, going through here. And then this is the larger bile duct with a columnar epithelium instead of a cuboidal epithelium. And plenty of collagen surrounding most of that triad group. Hepatocytes and sinusoids can be seen up here and down in this corner over here and a little bit up here as well. A small portal triad example. This is our arteriole right here, small artery right there, a branch of the hepatic artery. This is a part of the hepatic portal vein. This could also be part of the hepatic portal vein branch right here, so we have a couple of those. And we have a nice ring of cuboidal cells making up the bile duct portion. There's not a lot of collagen and connective tissue around that, but a little bit. So we can kind of see a very definite region for the portal triad, and everything outside of that is the hepatocytes and sinusoids and associated tissues. Another portal triad example. We have a couple of arteries here. One right here, it's a little bit larger, and a very small arteriole down here in the group. A large vein, portal vein there, hepatic portal vein branch. And here we have a bile duct right here. And collagen and fibroblasts surrounding most of this, making this outline around here. Everything out here, these are hepatocytes to the outside of the connective tissue. In this view, I'll point these out, just a couple of small bile ductules. There's one here, there's one over on this side, over here. So some bile ductules showing up there. We'll look at more of those later. One more example of a triad here. Here's your artery component. Here's the hepatic portal vein branch, very large piece of this. And then this is a fairly large duct. We've got columnar cells in this bile duct. And there's a smaller duct down here with less, not as tall cells, I should say. A cuboidal epithelium here, a columnar epithelium here, the artery branch, and the hepatic portal vein branch. Lots of collagen here surrounding this and fibroblasts there. This lighter piece right in here is another one of those small bile ductules surrounded by cuboidal cells. It would be a smaller duct than what this is right here, coming up into this general region of the portal triad. Next, we're looking at hepatocytes and sinusoids, endothelial cells and Kupffer cells in these examples. So the larger pink cells that make these continuous kinds of loose strands through here, these linear pathways, these are hepatocytes. It's common to see two nuclei together in a hepatocyte. It's a very common thing to see, a double nucleus hepatocytes throughout the tissue. The white space represents the sinusoid. This would be an endothelial nucleus. They're generally more flattened. Here's another down here. It's a little bit hard to see in this one, that space of Dissy between the endothelial cell and the hepatocyte. We see a little bit of it right here in this example. There are some Kupffer cells. Here's a Kupffer cell bulging out into the sinusoid space right here. This is likely one here, but not a good example of a Kupffer cell. So anyway, plenty of hepatocytes, sinusoids, endothelial nuclei, some Kupffer cells visible in that view. Now the next view, next example. Here we have a couple of sinusoids cut in cross-section. So this is a sinusoid here. You can see the endothelial cells. You can see a space of Dissy outside here. These are hepatocytes that surround it all the way around. You see hepatocytes. Another example here. So you see an endothelial cell here, some blood cells right inside that sinusoid. That gap between the endothelium and the hepatocyte is that space of Dissy. So we have a good space of Dissy here and a good space of Dissy around that example right there. Down here we have a sinusoid that's kind of collapsed so that the two sides of the wall of the sinusoid form this line down the middle, and the space to either side of that line would be the space of Dicey in this example here. Now this circle right here is one of the small bile ductules. Right next to it is a smaller one here and another one over here. So there's some bile ductules, hepatocyte, space of Dicey. Don't see anything that's certainly a Kupffer cell, although that could be a Kupffer cell right there in that section. Very likely is. Here the arrow points to an endothelial cell nucleus, a flattened nucleus. You can see the hepatocyte nuclei are nice and round. That space right through there would be the sinusoid space, and you can see a nice straight line. Straight line comes up to the nucleus of the endothelium right here. So anything between that straight line there and the hepatocyte would be space of Dicey, again, outside of the endothelium. This is an example from a slide that is specially prepared to show Kupffer cells. It's been exposed to a carbon material that the phagocytic cells would gather up prior to the preparation of the tissue. So these cells, when they were alive, they were eating up these carbon particles that were going into the liver. They acquired enough of it so that they retain it and have this very dark color in this slide. So all these dark patches you see in here are bits of Kupffer cells that have retained that carbon that's been put into the tissue. So that makes it hard to see the individual Kupffer cells for all that stuff that's in it. There are a couple of endothelial nuclei down here that obviously don't have the accumulation of the carbon material that was put in there. Here you can see the nucleus of a Kupffer cell as it's surrounded by that material. Here's another one where you can see the nucleus of the Kupffer cell, and then around that you can see its cytoplasmic region full of the black color. It's a nice big hepatocyte with two nuclei right here. There's a single nucleus there. So anyway, these are hepatocytes. Right in there is a bit of a biocanalicculus, but we'll find better examples of spots between the cells there where we accumulate the bile. We'll look at that later. Here's another one right here. Another view showing, this is to show the endothelial nucleus here. Sinusoid spaces through here. There's a Kupffer cell right here in the section, another over here. So anyway, we have the sinusoid, we have the endothelium, we have hepatocytes, Kupffer cells, space of Dicey. You can see pretty well over here on this side, that white space along there. We have some Kupffer cells. Here's a good example of a Kupffer cell in a sinusoid, the white space being the sinusoid. That's a nice example of a Kupffer cell right there. Not much of a white space here between the endothelium and the hepatocytes, but there's a little bit of a light line right along here that we can see in that location. Bit of a hepatic portal vein branch over here with collagen coming up to the hepatocytes. Looks like another Kupffer cell in a sinusoid way down here. So again, hepatocytes, sinusoids, Kupffer cells, endothelium, and space of Dicey. Things we can see in this view. This is a little interesting. There's a blood cell type right here, a white blood cell, next to a Kupffer cell right here. So the arrow is pointing to the Kupffer cell. You can see the endothelial lining, the darker line there, and then a little bit of a gap between the endothelium and the hepatocyte cells, the pink staining cells. So right in there would be your space of Dissy. Another sinusoid here, endothelial tissues forming that line, and then a little gap between that and the hepatocyte, forming a space of Dissy again. We're going to see more of the same thing over and over again. There's a good endothelial nucleus there's one up here. This cell right in the middle, though, right there, has a nice shadow of cytoplasm around the nucleus, and it's in a sinusoid space, so that's a Kupffer cell. There, that's what you look for. You look for a cell in a sinusoid with a shadow of cytoplasm around a nucleus. It's kind of an irregular shaped cell. So once again, we see hepatocytes, sinusoids, endothelial cells, Kupffer cells, and space of Dissy, all in that view. Repeating the same thing over again. Here we have, there's an endothelial nucleus right here, and a good line of that endothelial border around the sinusoidal space. There's your space of Dissy outside of that, and then the Kupffer cell. This slide was labeled for Kupffer cell, but I'm not seeing one near the center of the screen, at least in this case. So we'll skip looking for that and move on to the next one. This one also labeled for Kupffer cell. This one is a Kupffer cell right here in this picture, and you can see the endothelial lining, the space of Dissy, and the hepatocyte. Here's another, this is a sinusoid, this is a sinusoid. There's a tiny little sinusoidal space there. Anyhow, endothelial nucleus here associated with that sinusoid, and in the middle here is a Kupffer cell. This is a biocanalicus, a bile ductule, that's what I mean to say, a bile ductule right here, that circle of cuboidal cells. Notice that they are pale staining. There's a light area of cytoplasm around the nucleus, unlike what you see with a hepatocyte, where there's a nice pink staining nucleus around, cytoplasm around the hepatocyte nucleus. So again, we have sinusoids, we have endothelial cell, we have a Kupffer cell, we have plenty of hepatocytes, we have a little bit of the space of Dissy showing up outside of this, especially between that nucleus and the next hepatocyte, that white area across that side. And we have a bile ductule. Another example, there's sinusoid, a nice sinusoid space, good endothelial nuclei along here, and a little bit of a gap between the endothelium and the hepatocyte, showing there the space of Dissy in here and up along this side here. Another bile ductule right here and right here. In this particular slide, our space of Dissy is kind of exaggerated and our sinusoids are kind of collapsed. So in here would be our sinusoid space. has a little bit of blood in it, and some Kupffer cells. And anyway, so that's large white space next to our hepatocytes. That is the space of Dicey on either side there. There's an endothelial cell right here, and that space between the endothelial nucleus and the hepatocyte, that white space, would be space of Dicey. This, I'm not sure if that's a Kupffer cell or not. It's kind of a small nucleus, but there's a shadow of cytoplasm around it. I wouldn't use that as an example, but possible Kupffer cell in this sinusoid. This would be sinusoid space, and so that white space above that, still space of Dicey in there next to the hepatocytes. This is to show the central vein and the radiating sinusoids. So we have a nice central vein space here. We have some endothelial nuclei, small dark nuclei. This is a lower magnification. And we have the plates of hepatocytes. You can see them radiating out. Lines of these larger pink cells with a nice round nuclei. And then the white spaces being the sinusoids that travel out away from that center. So we have a few sinusoids connected to that central vein in this example. Another central vein. Again, you see this radiating pattern, lines of sinusoids that radiate out away from the central vein. There's a fair amount of blood in that, red blood cells scattered around in there. There's some endothelial nuclei there. Here's the connection of a sinusoid coming up. This was the slide that had the Kupffer cells with the black. So all those black spots represent Kupffer cells in the picture. Nice lines of radiating hepatocytes. Same thing again, a central vein in the middle, and you can see sinusoids connected to it and the hip plates of hepatocytes that radiate out from the middle, from that central vein. Remember that at the perimeter of this lobule, this liver lobule, there would be some of our triads, our liver triads. Here we're looking for bile canaliculi, those small passages between adjacent hepatocytes that will conduct the bile. So bile gathers into these spaces. Right at the end of the pointer, there's kind of a fuzzy dot there. But over here we see a short segment of a line between adjacent cells where the bile would accumulate there. And that's a small bile ductule right there. I think I had some other locations on here. Let's see. Right here, there's a bile ductule. I'm saying the wrong word, sorry. Bile canaliculus. That's what I'm supposed to be saying. This is a bile canaliculus. That's a bile canaliculus, and this is a bile canaliculus here. There's an example right there, a fine example. That little spot is a bile canaliculus. So bile accumulates right there between adjacent cells. There's a 2 nucleus hepatocyte and a 1 nucleus hepatocyte, and the bile canaliculus between. There's another good example up here. Again, that slide that has the Kupffer cells with accumulated carbon particles in there to make it dark and black. And here between this cell and this cell we have a little oval right there where bile accumulates between adjacent hepatocytes, the bile canaliculus. There are a few others on this example. There's one right here and a good one right there, a little white dot right there between those cells. Bile canaliculi. Bile ductules we've seen in several of our slides already. And remember I said they looked like a stepping stone arrangement in cells that had a clear cytoplasm around the nucleus. The cells are smaller than the hepatocytes, and they don't have that dark pink stain around the nucleus. So they're a different looking cell. So sometimes they look like a strand, like that stepping stone pathway look, and sometimes in cross-section they make nice circular patterns. Here's one of the tiniest ones I've seen. It just has a couple of nuclei there. But anyway, there's another example right up here. Down over in this corner, there's a nice circle of cells down there. It shows up on the screen. I think we used this somewhere else in the same example here. It shows the nice endothelial cells, the space of Dicey and the sinusoid. Another bile ductule there in the middle of the screen. You see that line of cells with the smaller cells with light staining cytoplasm around them, headed up through here. Another example, remember the light spaces are mostly sinusoids out through here, and then the pink cells are the hepatocytes, and right along here is a nice long strand of that stepping stone pathway with the light cytoplasm. Small cells, that's your bile ductule again. Okay, and this view is not a real great view of most of the liver tissue. You can see a lot of hepatocytes and some sinusoids in here and a few things, but there's a lot of lack of detail. There's a nice Kupffer cell, I guess, right in there. But this shows fat cells, adipocytes, among the liver cells, which is not something you commonly see in these slides, but you run into it occasionally, and this is an example with fat cells in the liver. And this will be our last slide. This is the large hepatic duct. A large hepatic duct. It's got a columnar epithelium along the wall. It's got a thick layer of connective tissue to the outside of it. This would probably lie very close to a large portal vein and a large artery passing through the liver. But anyway, this is a section of the large bile duct. You can see all that huge space in there, very big duct, and nice columnar cells along the wall. A large hepatic duct.